Next up, we have Emmanuel Amewu from School of Medical Sciences at Nus University in Ghana. And the title of his talk today is The Association Between Submicroscopic Plasmodium Infections and Gut Microbiota Among Children in Ahanta West, Ghana. Welcome.
Hello, everyone. Can you hear me?
Yes, we can hear you all right.
All right, thank you very much. Hello, everyone. Good afternoon from Ghana here, and thank you, BSI, for the opportunity to present on my work on submicroscopic Plasmodium infections and gut microbiota among children in Ahanta West, a malaria endemic community in Ghana. Fortunately for me, Dr. Mota has done a lot of the groundwork related to this work on their work on Plasmodium infections, the liver stage. Now, I won't bore the conference attendants so much with the nitty-gritties of the malaria disease. However, we know currently that about 300 million cases of malaria is reported globally, and there are about almost 600,000 deaths. And out of this mortality and morbidity, 90% of this burden lies in the African region. And out of the morbidity in the African region, 3 out of 4 of them are children below 5 years of age. And this is very troubling with all the advanced work in the vaccine production and all that. We are still not out of the woods because of the ripe climatic conditions that the mosquitoes enjoy in our region over here. Now, malaria transmission is key to the disease itself, and most of the time, the part that is neglected are the submicroscopic infections or the asymptomatic and submicroscopic infections, which present themselves some diagnostic challenges with the case of malaria. In Ghana, since 2019, a few studies have looked at submicroscopic Plasmodium infections in children, and they reported a prevalence ranging from 27% to about 47% in the region. And there are so many risk factors that have been associated with it, which include the ripe climate and then the year-round rainfall that sustain all of these things. However, there is a new branch of medicine, the human microbiome, that has been linked favorably with a lot of conditions, including infectious disease and non-communicable disease alike. Recently, there have been some studies that have looked at the possible interactions between malaria and intestinal parasite infections and the gut microbiota. There have been some studies in Ugandan children and also in Malian children looking at the association between gut microbiome with severe malaria and also the febrile or susceptibility to febrile malaria disease. However, the aspect of asymptomatic cases that elude the mainstream clinical cases are not really looked at, and that was the focus of my work over here, to look at the focus on the limited gut microbiome associated data from endemic regions with the asymptomatic and submicroscopic Plasmodium infections. And of course, there are fewer submicroscopic studies or data on them because of the nature in which the disease are. And Plasmodium species-specific effect on the gut microbiota also remains unclear if different species of the Plasmodium elicit different influence on the gut microbiota. So for this study, this is what we looked at to determine whether there was any association between the gut microbiota composition and then the Plasmodium infection among school-age children. So specifically, just 3 specific objectives: to look at the prevalence of submicroscopic Plasmodium infections, look at the gut microbiota composition of these children, and then try to evaluate if there is any link between these two. This was the research approach. We decided to go with a cross-sectional exploratory to find what is there to inform future studies. It was a community-based survey at first among children in Ahanta West, and we collected data on their demographic, medical history. We took venous blood samples to diagnose malaria or Plasmodium infection, and stool samples for gut microbiota analysis. For the malaria diagnosis, we employed microscopy, RDTs, and multiplex real-time PCR to detect the various species of Plasmodium that were present in our study. And we went on to do the variable region 4 sequencing of the 16S rRNA gene. And we employ both bioinformatics, biostatistics in this analysis. For the characteristics of the participants, majority, almost 2 out of 3 of our participants were females, reflecting the gender distribution at that age. And most of the participants were also between the ages of 9 and 12, and reflected in their education status, with about 1 in 3 in high school. Majority of the participants had dewormed before the sample collection, and a few of them reported taking any antibiotics or anti-malaria. Now, on the outlook of our first objective, we saw that for the submicroscopic infections, there was 53.1%, which we consider to be high, considering that the microscopy itself in the middle section, we encountered 30.1%, and there were mixed infection, Plasmodium falciparum, and then mixed infections of Plasmodium falciparum and Plasmodium malaria. And for the PCR, there was over 77% positivity, and majority of this infection was from Plasmodium malaria, and we had some mixed infection from Plasmodium falciparum and Plasmodium malaria. And the RDTs, we used 3 different test kits, and we saw consistently some level of consistency over there between 44.2 to 52.2 positivity with RDT. Now, going on to the gut microbiota distribution amongst the participants. Initially, we grouped the participants into those that were negative and those that were microscopy positive, and then those that were PCR positive, microscopy negative for the submicroscopic infection. And generally across board, we saw that there was reduced richness and evenness in the infected group compared to the negative, although the general distribution was not statistically significant. And looking out for if there were any peculiar or clusters of microbial communities that were distinct between the groups, there were just overlapping circles indicating that most of the participants shared a common microbial population. However, there were some that were differential, or we could see that were outlying, and we went on to do the differential abundance to find if there were any significantly enriched or depleted microbial communities in our study participants. So between the asymptomatic, those that were positive for microscopy, and then for those that were submicroscopic based on the PCR, in both cases, we saw that there were some enriched and depleted microbiota based compared to the negatives. Now, in this graph, those on the right of the dotted line represent the enriched, and then to the left, we have the depleted. And we can see the enrichment of Clostridium species, which have been associated with some broad range of invasive infectious infections in humans, and even associated with colonic necrosis, was enriched in the infected group as well. And then for at the bottom, we see Cicatella, which is a predominant the asymptomatic. It was a similar trend comparing the asymptomatic to the negative as well, where we had a reduction in microbes that are thought to colonize largely the gut, and the enrichment of what has been associated with some inflammatory state in other conditions in other cities as well. And some of the outlook of area, like the Zalanibacter, as we see here, may present the plant-based nature of the diet of our study participants. However, there's also the enrichment of Klebsiella, which is of clinical interest, especially considering the escape pathogens group. Now, we wanted to see if the Plasmodium species that are causing the infections have any specific influence on the gut microbiota, given that we saw a lot of Plasmodium malaria infections from the PCR. Over here, generally, there were reduced richness and abundance, looking at the Shannon diversity for the infected groups compared to the negative. However, the chart one that looks for rare organisms in the microbiota, we see that in the mixed infection, which are those with the Plasmodium malaria and Plasmodium falciparum, had higher richness, which may indicate some peculiar or some distinct microbial signatures in the infection states, and engenders further studies to identify some of this. And in this regard, also for the beta diversity, to look at the clustering of microbial communities between the groups, we didn't find any significant clustering, and we went on to look at the few that we saw or we thought that were differentially abundant or depleted. And over here too, comparing the participant with mixed infection to the negative, and then the Plasmodium malaria only with the negatives, they were also enriched and depleted microbes that were of interest. We went on to extract them to find their biological or clinical relevance. And over here, what we see here generally is the depletion of Cicatella and other microbes that are known to colonize or to be predominant in the gut microbiota as well, and then the enrichment of Treponema and other bacteria that have been associated with some sepsis and infections in humans and even in swine. It was a similar outlook comparing the Plasmodium malaria versus the negatives. Also, we saw the depleted microbiota from those that have been thought to be beneficial, and then enrichment of gut bacteria that have been associated with inflammation in other studies. Now, finally, we wanted to look at the influence of all the variables in our study, some of the selected variables in our study on the gut microbiota with the redundancy analysis. And from here, the general model, the universal model, was not significant. However, specifically the Plasmodium species causing the infection had the longest arrow, which was also significant, and it shows the influence of the Plasmodium species causing infection on the gut microbiota. And deworming as well also had another influence, probably pointing to some competition in the gut, as they are all in a common niche inside the human. So in conclusion, we saw some high prevalence of submicroscopic Plasmodium infection, which is of concern in breaking the transmission cycle and bringing malaria under our control, and potential emergence of Plasmodium malaria, mostly beside Plasmodium falciparum. We have focused a lot on vivax and ovale, but this could present another challenge that we have to look at and nick it in the bud. And the next is the low microbial diversity between the infected and the uninfected. Although the models were not significant, there were some enrichment of some gut microbiota that have been associated with pro-inflammatory states. And this could also point to potential infection specific or Plasmodium specific microbial signatures that could be capitalized on for the development of therapeutics or in diagnosis to help in this fight against malaria. I would like to acknowledge my supervisors, Dr. Malabi, Prof. Kwatin, Prof. Yabua, Dr. Iwa, and the research team that went to the field and did helped in this study, as well as our study participants as well. This study was supported by the UNESCO TWAS grant, and we had a lot of support from the Microbiota Vault, and the University of Zurich for the microbiota sequencing of our samples. These are a few selected references. Thank you.
Thank you so much for a wonderful talk, Emmanuel. I think this is especially relevant after our keynote speaker, Dr. Maria Mota today, because her talk was also on malaria to some degree. Yeah. A question from our audience is, over time, do you observe that the microbiota signature changes and influences disease inflammation or sterile immunity?
Well, a very important question. However, in our study, this is cross-sectional, so we didn't follow the participants over time. So it will be difficult to answer that. It's difficult to say or to point the direction if of the subtle association, if I would say that we find whether it is the infection state that leads to the dysbiosis or it is the dysbiosis that makes the individual susceptible to the Plasmodium infection. It is something that we hope to look at in the future.
Great. Another question from our audience. How does the Plasmodium gut microbiota affect susceptibility to other pathogens? And do you suspect that microbiota shape the immune system?
Yes, so I think there have been a lot of studies that are pointing to that, especially learning how some of the immune cells are trained and mature or are trained by the gut microbiome. So yes, there is a strong link coming up and a lot of work points to that. And yes, as I indicated earlier, it is a risk factor or one area that is gaining a lot of attention in recent times because in many diseases, like even in cancers, in neurodegenerative diseases, it has been linked. And we can see that, I mean, considering the number of cells in the human body and the number of microbes, we can say we are more microbes than ourselves. So the link is possible, and I'm sure we'll find more.
That's great. Another question from our audience. Have you anytime considered incorporating biotics to your work? Would love to hear your views on that.
Adding biotics like an intervention for the participants.
Yeah, Sukandra, if you can clarify your question in the chat, that'd be great. Um,
Yes, so I think one thing about the probiotic and prebiotics intervention
yeah.
as a dietary intervention, because one of the things that we considered was the nutrition status. If we considered malnourished and normal children, how is this also like? And it is something that we are working on. But sure, of course, nutrition is known to impact immunity and immune response. And it's more of a systems approach. It's difficult to look at these things in isolation. So we are considering looking at all of it together to link them very well.
That's great. Another question about experimental design. Is there a particular reason why you picked this specific age group? And do you think age stratification um, reveal associations that were not apparent in the full cohort, or would extending the age um, reveal something else?
Yes, so there are two sides of the coin here. So malaria susceptibility and how it affects individuals in endemic regions like in Ghana are different. So for children, it's more severe, and because of their immature immune system, we tend to see what do you call it, more severe cases in them. However, as you age, because of exposure and over time, adults tend to have tolerance and build some level of immunity to the disease. And it's similar thing also for the microbiota from the gut, as it also develops once, I mean, you get inoculated from birth, and as you age, as you eat different kinds of food, expose yourself to the environment and the interactions, you also build and have your gut microbiome maturing to some stage, which can also change or be modified with diet and changes in environment. So there are a lot of factors or confounding variables that can impact, I mean, the gut microbiota. Even if we look at just medication, diet, and I mean acidity, from the gastric juices and all that. So all these are possible to influence the outlook of the interaction between the infection and then the gut microbiota.
That's great. Eleanor, do you want to ask your question?
Sure. So that was an excellent talk, um, and it's really interesting. But I guess I have a couple of questions. So, children are born of mothers, and many of these women in endemic regions have had exposure to malaria over decades, repeated exposures. And we also know that in utero, there's exposure of the fetus that influences the immune system of the child. So it's certainly that will shape them, and we know this again that vaginal birth shapes the microbiome. So my question to you is, when you're studying these children, what kind of histories do you get on their mothers, on what their exposure, and how soon after birth these children might or might not get malaria? How I mean, this is a huge undertaking. How simple is it to do, knowing that many of them probably are in rural regions? If you can just give us a little bit of a sense of how you go about, collecting these patients, getting these histories, and trying to pull all of this together as you shape your hypothesis and build your experimental design. That'd be really interesting for all of us.
Yes, so this is a very interesting angle, and it's a huge conversation that we've had in the past with especially the health directorate of this particular district in which we did this study. Now, as you indicated, mothers are exposed repeatedly and through the development of the babies until birth. Now, in these endemic areas, one interesting thing we find is that there is what we call the low grade, low parasitemia amongst a lot of the children that we find over there. And one report which we happen to look at is that they tend to have a lot of cerebral malaria cases. And it's interesting to find that when they develop malaria, it's severe. And when they do the parasitemia, is usually very low. We live in this region, we know it's endemic, it's hyper-endemic. However, we don't really get a lot of the children coming with the severe or coming with the febrile disease, but they come presenting with cerebral malaria. And when we test them, the parasitemia is low. And it showed the dynamics of how the immune system of the mothers are trained, and if there's any level of immunity passed on to the children, and as they develop, or as they now for if I can paint a picture of some of these communities, they are end of the road kind of communities, and should I say villages that have limited access to healthcare facilities. And in most cases, when there are these interventions for treated bed nets to prevent some of these things, it's probably there's the psychology of the people. We go in and we find that these are used as garden fences, or complain about the heat when they sleep in the mosquito nets and stuff. So it's complicated, but as I indicated, probably there may be more to this, and that is why we decided to explore with this. And given the nature of the data that is coming out, there are other individuals in the lab that are also analyzing different components of the study as well.
Thank you.
Thank you so much for your wonderful talk, Loba.